Good morning, everyone. Welcome to the final webinar in our 2021 Winter Science Seminar Series, brought to you by the Southeast Climate Adaptation Science Center, otherwise known as SE CASC or Southeast CASC. My name is Carrie Furness, and I'm the program manager here at the Southeast CASC. In this virtual seminar series, we've been highlighting Southeast CASC funded projects that support resource management actions across the Southeast. So just to give you an idea of what to expect from today's webinar, we'll go over some quick meeting logistics, request some feedback via a short poll, and then we'll introduce our speaker today. He'll present for 40 minutes or so, and that'll give us some good time for Q&A at the end of the seminar before we wrap up. So I have a nifty Zoom basics slide that I was going to show you, but going on the premise that you all are Zoom aficionados now, I'll skip that. But I did want to quickly cover some of the features, since I know some of you use other remote technologies. So the controls at the bottom allow you to mute and unmute your audio, ask that you keep your audio muted. We will mute you if necessary, just to reduce distractions, and to also keep your video off during the presentation, just to reduce distractions for our presenter. So in the middle of the bottom bar, you can see a chat window where you can please ask questions, and encourage you to throw questions in there during the course of the presentation so that we can entertain those during the Q&A session. We are also recording the presentation today, and you can access and share the recordings later on our YouTube channel. So we'll give you that information at the end for where you can access that. So now I'm going to pass the mic over to Ranzizu Laskarain, our Assistant University Director here at the Southeast CASC.
Good morning, everyone. Thank you for joining us. I'm just going to launch a quick poll, just 2 questions, just to get a little information about who is with us today and help us know how to continue to get information out about these seminars and to make sure our partners know about them. So we'll just give you about a half a minute or so to fill it out, and then I will share the results with you all. And I'm going to launch the poll now. All right. Fantastic. Excellent, excellent. Just a couple more seconds. All right, I will end the poll and share the results with you all. That's terrific. Yeah, we're about equal on federal agency and university partners, some state agencies. That's fantastic. Always really pleased to get NGO folks and community members, of course. And one of the things that we've always been trying to find out is how you all learn about these seminars. And so clearly our newsletter, our monthly newsletter, is a really great source of information. So that's fantastic. Colleagues, that's great. Continue to share with your colleagues, please. A little bit on Twitter. Okay, good to know. Thank you so much for that quick poll. And now I will pass it back to Carrie. Thank you, Carrie.
Great. Thanks, Ranzizu. So now we'd like to move on to our presentation. And first, I'm delighted to be able to introduce our speaker, who is Dr. Ken Krauss. He is a physiological ecologist and wetland scientist at the USGS Wetland and Aquatic Research Center in Lafayette, Louisiana. Ken's research spans several habitats, from mangroves to tidal freshwater forested wetlands and marshes, and takes a multi-tiered approach to understanding eco-physiological processes in coastal wetland forests, defining gas exchange between the soil and atmosphere and among the leaf, tree, and atmosphere. His research has defined thresholds to tidal freshwater forested wetland habitat change in the face of persistent environmental drivers, specifically sea level rise and salinity, defined the potential of forested wetlands to influence water cycling in coastal areas, and has begun to establish the potential of other wetland types to contribute to water conservation, especially under drought and perennial salinization. Ken also focuses on the vulnerability of coastal swamp forests and mangroves to sea level rise, and on how science can inform management and restoration activity within the coastal zone. So Ken will be discussing today a great research project that was funded by the Southeast CASC, as well as some other USGS groups. And the title of his project and the talk today is Science to Inform the Management of Mangrove Ecosystems Undergoing Sea Level Rise at Ding Darling National Wildlife Refuge in Sanibel Island, Florida. So now I'll turn it over to you, Ken.
Okay, well, thank you very much. I'm assuming you can all hear me. Beep in if you can't. I really appreciate everyone taking the time to listen to this presentation today. It's the first time we've been able to put together what's become the story of Ding Darling National Wildlife Refuge. And I do want to apologize if I have a sausage dog fly out of the bed and make a lot of noise. And apparently it's squirrel season currently. She goes crazy every once in a while. Okay, so I wanted to start out by saying that I'm the lead principal investigator on this project. And I was thinking about it the other day, like how much time I actually spent in the field. Probably very little, probably like 3 or 4 trips. Most of the science that I'm going to talk about here has been conducted by the project scientists and the students listed here. And I also want to shout out to Dr. Z, who was the original coordinator of this particular program through Land Carbon. And I'll speak more on that later as I go through this presentation. So I should be advancing. Okay, there we go. I'm hoping you see the structure of presentation slides right now. Okay, great. Yeah, we had issues with advancing earlier for those of you who are wondering why I said that. So I'm going to go through this. I want to give an introduction to Ding Darling National Wildlife Refuge and sort of introduce the concern that we had 5, 6, 7 years ago, however long it's been at this point. I want to remind everybody about the vulnerability and how sort of mangroves can be vulnerable to sea level rise. And this is a review for everyone. And I'm going to be quick. I'm not going to do it like I normally do it. And most of the presentation is going to be about our approaches and summary of the results. And then I'm going to talk a bit about the future of Sanibel Island's mangroves. I'll mention that Ding Darling National Wildlife Refuge is located on Sanibel Island. So that's why you'll see me jump between the two seemingly different locations. They're the same location. I gotta remember. Okay, so this is Ding Darling Refuge. This is the map you receive when you go to any kind of visitor center there. And it shows you the roads, it shows you the birding, places to go birding. It shows you roughly where the mangroves are, all the dark green, and who to contact if you see something wrong. But what's interesting is where this island actually sits. So here's in this inset, you can kind of see in this lower left-hand side where Sanibel Island sits. And look at this river, Caloosahatchee River. This is one of the few rivers in Southwest Florida that have fewer regulations, or I don't want to say it's unregulated, but it isn't regulated to the degree that other rivers are apparently. And you can kind of see a lot of these straight canals. These are associated with human development, so agriculture, things like that. So you can imagine that this river can take a lot of runoff. And we'll talk about what that runoff looks like, but I do want to mention here that one of the big concerns for the mangroves at Ding Darling is the increased nutrient loading that has been coming down the Caloosahatchee River. This is sort of the same story that's been unfolding when you see the algal bloom. The manatees in Southwest Florida are dealing with this now, where increased nutrient loading are causing algal blooms that are shading the water and killing the grass. Therefore, the seagrass, therefore, manatees have a hard time surviving. Many are dying now. It's the same story, it's all interlinked. And it just causes concern for these mangrove forests on Ding. So this kind of was the beginning and the genesis of our project hypotheses in the beginning. Okay, so the refuge, named after Ding Darling, J. Norwood Ding Darling. He died in 1962. He was the first chief of the U.S. Fish and Wildlife Service. He was a cartoonist, avid conservationist, very impressive guy, and he, of course, did a lot in Southwest Florida. This is considered one of the best places to birdwatch. People from all over the world, really, but certainly all over the nation, come down to bird watch. I'm not a bird watcher myself, but I hear it's a phenomenal place to go. The Refuge was established in 1945, and it has about 2,500 hectares of protected area, open water, beach strain, estuarine habitat, and a fairly large, about 1,000 hectares of mangrove forest, give or take. All of these pictures were taken on the Refuge except for one, but I will tell you that what's interesting too, this picture of an iguana, even some animals that might not be totally welcome are around as well. And I gotta say this, one of the things I really enjoyed about working at Ding Darling was being exposed to this series of books by Randy Wayne White. He writes about sort of the culture of the area, and he bases everything basically on Sanibel Island. He calls it something different in the book, but it's a really good read, and he's written many of these books. The first one is Sanibel Flat, but it was really inspired by Ding Darling National Wildlife Refuge. Okay, so the first thing we did was actually pre-land carbon project. Again, I'll mention to you what I'm talking about when I say land carbon in a second. And this was done by Ellie Reed. And what she did was she came in and she wanted to look at the carbon standing stocks at Ding Darling National Wildlife Refuge. Many of you realize that, I mean, one of the biggest stories in mangrove ecosystem ecology right now is of course carbon. So the first thing she wanted to do was map areal change and map recovery after a major hurricane. So what she did was she came in, gotta learn how to advance, and she did a series of imagery analysis using the Landsat imagery from 1985 to 2015. Initially, we were going to continue this analysis into current day, but that hasn't happened. About 1,000 Landsat images were used to do this analysis. And what she discovered was there's about 1,000 hectares of mangroves in Ding Darling, but in 2004, there was a drastic decrease. And of course, this happens when you get hurricanes. So Hurricane Charlie came through, Category 4 storm. It really slammed Ding Darling. And as many of you are familiar, probably, is that mangroves are often touted as being disturbance adapted communities. So they do have an adaptation. They do adapt if they're healthy. They have the ability to adapt and recover from these storms. But one thing we noticed is 10 years later, the recovery wasn't altogether there yet. It's still trying to recover. So we were a little concerned with this lack of recovery. And I will mention that a couple more times throughout this presentation. Okay, so I wanted to sort of just remind people of what sea level rise and what can happen when, in terms of mangrove responses to sea level rise and what can affect that response. So what we wanted to do is define the degree to which natural habitat persistence and carbon vulnerability of mangrove ecosystems at Ding is influenced by sea level rise, and in particular, what is the role of the ongoing nutrient loading down the Caloosahatchee River? This was our primary concern. And just to remind people, so mangroves have sort of two ways of responding to sea level rise, one of which a lot of people talk about is this migration inland. Migration inland can occur if the system's healthy. The forest does have to be gaining elevation. It can't be less than 0. But if it is gaining elevation, it can indeed move inland if the slope of the landscape is sufficient and there's no barriers such as roads or seawalls. You hear a lot about this. But what I really wanted to do is mention this part of it, which is remembering that mangroves have an ability to adjust vertically to elevation. They don't just sit there and submerge. They actually have this feedback mechanism that allows them to build. There's a limit to that ability to pace sea level rise. And I won't go through the processes here, but there's a lot of processes that mangroves can use if their habitat is healthy. And so our critical observations was, of course, I mentioned this, that recovery after Hurricane Charlie seemed a bit slow. We were a little concerned that maybe this isn't a natural trajectory. I will say this too, that many places in South Florida are being influenced by anthropogenic change. The more we look at this, the more examples we come up with of change that's affecting mangrove systems in Southwest Florida. It's everywhere. And of course, our concern about river nutrient loading. We're worried that maybe it's creating a slowly progressing vulnerability to natural sea level rise within the mangrove at Ding Darling that you really don't notice just by casual observation. Okay, so how can humans affect response at Ding Darling National Wildlife Refuge? Well, there's really two ways that come to mind. So one of which is altered hydrology. Many of you know that Ding Darling has a history of impoundment management. All in all, the study area here isn't focused on impoundment. It's focused on the natural tidally influenced mangroves at Ding Darling. And I'll mention this, that if you look at this hydrograph at the top, this dotted line here is your soil surface elevation versus the hydrograph on the bottom. And I don't have units on the y-axis, but about 30 centimeters or so probably. That there's a lot of hydrographs. There's a lot of mangroves that have this hydrograph on top throughout the state of Florida and globally. But this is created by modifications such as roads, not so good culverting. There's a lot of things that can cause this. So this is one of the biggest drivers, and I'm showing you this here because of all the drivers that we look at, altered hydrology is the biggest. And in particular, what we're worried about is this. This is the process of peat collapse. It doesn't take much to make a mangrove unhealthy, and it doesn't take much to force them to start losing surface elevation over time. You don't notice it. It takes many decades sometimes for people to start noticing this change in mangrove systems, which is why it's not ameliorated early on. But basically what happens is if you look at a soil core, and this is bulk density, so this is density into the soil. The green represents roughly the root zone, so it stops at about 40 centimeters. This is not a healthy forest here, but it's a healthier forest. What will happen is as this root zone, if you look at this particular graph versus this graph, the root zone loses elevation because the roots die, decomposition is higher than the new root growth, and you get this zone of compaction that allows for this soil to slump down, basically. And this wasn't from Ding Darling, but it was from Marco Island, which isn't that far away from Ding Darling. And you lose this surface elevation, 10 centimeters of surface elevation loss is a big deal. And one of the drivers of peat collapse, besides altered hydrology, can be nutrient loading. So Ding Darling National Wildlife Refuge, basin mangroves have up to 4 times the phosphorus concentrations of other mangroves in Florida. And this is a big deal. We were super shocked when we looked at the total phosphorus concentrations in the soil. And I will tell you that nitrogen isn't necessarily distinctive from other forests in Southwest Florida, but phosphorus is super high in the soil. So we thought this has to have some kind of consequence. So this research began as a USGS land carbon program project to investigate the role that management has in facilitating carbon loss or gain within natural wetlands within the National Wildlife Refuge system. So this is just one location that the land carbon program has focused on. This is the Mangrove Hub, as we refer to it as. There's other projects at Great Dismal Swamp National Wildlife Refuge and the Squally National Wildlife Refuge. There's a focus in South Louisiana as well. So there was one false defunding period, and then there was in the last administration, land carbon monies were pulled, and we were fortunate enough that the Southeast Climate Adaptation Science Center stepped up and paid for the last 2 years of the work and allowed us to ramp it down. So we really are appreciative of that. So how are we studying this factor, this process? So this is the Caloosahatchee River, and I don't know how many of you can see it's written Caloosahatchee River with the arrow. It's in black. I should have changed the color of this, but this is Sanibel Island, and there's a natural nutrient gradient from right all the way to left, and we thought a bit about this. We thought about using the natural gradient as our sort of natural treatment, but we decided that wouldn't be very impactful. We wouldn't really be able to link effects to specific nutrients. So we decided to undergo a fertilization event. So we actually took the middle site, right in the middle of this landscape gradient, and we established 3 blocks. Blocks were 250 meters apart. Each block had a fringe and a basin site. So fringe meaning the mangroves that are just on the estuarine edge, sort of the mangroves that you're used to seeing with the really cool roots and everything, the prop roots. And then we established basin mangrove forests just inland of that. A large percentage of basin mangroves in Southwest Florida are actually basin. They're not the ones you see from the boat. They're the ones that are behind the ones you see from the boat. And that was a big concern for us because a lot of wetland loss globally is of interior wetlands. So we established 3 complete replicates, so a total of 6 8 by 8 meter plots. And these plots were fertilized with granular nitrogen and phosphorus on a 2 by 2 meter grid with the idea that no fertilization application was more than a meter away from a plant, a tree, any kind of plant. And I'll mention this to shout out to Jeremy Conrad. This was part of his dissertation work, or is his dissertation work. He did all this fertilization. This is a lot of work in place. We had nitrogen plots, we had phosphorus plots, and we had control. So every nitrogen responses in the nitrogen plots, responses in the phosphorus plots were compared to the controls over time. He placed the fertilizer at 30 centimeter soil depth, and he did this twice annually for 3 years. So it was a lot of effort just to do the fertilization. And here's, so what we collected background data. We looked at above ground carbon standing stock. This was greatly facilitated by the work that Ellie Reed did that I showed you in the previous slide. We looked at soil carbon standing stocks and soil organic carbon accretion. This work was done by Judy Drexler with the USGS in Sacramento, California. And we looked at, so we also wanted to look at response variables specifically and directly to nutrients. So we looked at above ground and below ground productivity using dendrometer band and root ingrowth techniques. Both of these were are part of Jeremy's dissertation work. And then we wanted to look at ecosystem metabolism, soil and nematophore CO2 fluxes, photosynthetic water use efficiency, and sapflow. And then finally, and I don't know if you can see this, mine's covered, but I can move this thing. Finally, soil surface elevation change, because all of this for us is manifest, we think are the drivers potentially. I mean, the biggest question is, how is this system affected in its response to sea level rise adjustment? This is what keeps it there or not. And then we developed a couple of tools. We developed a sea level rise model, which is going to be kind of cool, I think, or it is cool. It's in the process of being developed, but I do have results to share, and an ecosystem services assessment. So all of these are to try to tell the story in a bigger way as we go. Okay, so the background data collection. So the mangroves at Ding comprise approximately 363 to 367 gigograms of carbon. This is total standing stock. This is about 363 billion grams of carbon. This is based on 2018 estimation. This is its distribution. So you have above ground, about 67 to 70 megagrams, a million grams of carbon per hectare. And then you have below ground. Most of the carbon is below ground, as is the case for 99.9% of all wetlands in the world, definitely for mangroves. You can see all the carbon that's stored below ground here. And this is my lame attempt to draw the roots. Yeah, I don't know if it's good or bad, but this is below ground. This is above ground. And so a couple of points. Again, above ground carbon increment is still recovering from Hurricane Charlie, but remember that that's going to have a feedback into root turnover below ground as above ground recovers. You expect more turnover below ground. And we may actually be seeing that now. I'll show you why. I think that in a second. Soil carbon burial rates from the basin mangroves were estimated at being, we could not estimate soil carbon burial within the fringe mangrove forest because of the turnover. We could not, Judy could not get good dates on using lead-210. This is a common problem in areas where there's a lot of turnover in a root zone. We'd like to try that again more later. But 0.61 to 1.3 megagrams of carbon per hectare per year, that's how much carbon is going into the soil within the basin systems. And this kind of matches what you see from the larger part of Florida, 1 to 1.6 megagrams. Josh Bright Hop showed this multiple years ago, and it's still within the range of these Florida estimates. So there's nothing real special here or real different here. But remember that one of the biggest concerns here is the vulnerability of all of this carbon, because carbon, though we focus on that, carbon represents the system. It represents the ecosystem and all those other ecosystem services that having that amount of carbon and structure associated with it, it provides to people, fish habitat, et cetera. So we're really worried about this vulnerability of the carbon. Okay, so What about response to nutrients? Now, this is where it gets exciting, or actually kind of interesting, not super exciting, but interesting. You'll see why I'm saying that. So past mangrove studies, past fertilization studies in mangroves have shown really important effects of nutrients. So if you fertilize a plot with nitrogen, you fertilize a plot with phosphorus, and sometimes there's a combination of nitrogen and phosphorus, and you compare that to controls. A lot of mangrove systems respond, especially to phosphorus in the Caribbean basin. But all of these studies were really in nutrient limited mangroves, so short stature, short mangroves less than 3 meters tall. We were curious about, well, what about an existing mangrove forest that's not stunted, that's 10 meters tall? And that's sort of the genesis of this. What does nitrogen and phosphorus fertilization do to that type of mangrove forest? Well, starting to show you some of Jeremy's dissertation work here is, I got to keep moving this little thing now, sorry. Installed dendrometer bands on all trees greater than or equal to 5 centimeters diameter of height per plot. So this is a dendrometer band here. So you can measure microfine increments in growth. And so the blue here represents fringe forest. The orange represents basin forest. So fringe control, fringe nitrogen plots, and fringe phosphorus plots. Remember that there are 3 of these, three replicates of each of these. And so what he discovered is that you look at growth increment, lateral stem growth is greater in fringe than in basin. Other than that, there are no differences in above ground growth. And this is after fertilization for up to 3 years. So that's, not super exciting. He also looked at below ground, Jeremy also looked at below ground productivity. He installed 6 root ingrowth bags per plot and extracted these over a period of 2 years. There's a delay in planting them, so he waited until a year after fertilization began. And again, not exciting. There were no differences in below ground productivity. Of course, many of you who do below ground productivity probably aren't too surprised. It's a hugely variable response among different treatments. So anyway, there was no difference there. Okay, so what about ecosystem metabolism? Because maybe it's that growth hasn't had enough time to manifest, to really respond to this nitrogen and phosphorus loading over 2 to 3 years, because it's only been going on for a short period of time. The extra loading has only been going on for a little time. So Natalie Ferrone at Florida Atlantic University looked at soil and nematophore restoration as part of her thesis. And this is what she found. So if you look at soil CO2 fluxes on the y-axis here, and she looked at ecosystem, at soil respiration in the dry season and the wet season, and of course, control nitrogen, phosphorus. And what she discovered was that the only real stimulation that she found in soil respiration was by phosphorus during the dry season. So not a whole lot to write home about yet. But here's what's kind of interesting to me. What she showed is that across all of these treatments, there was a response to increased soil temperature, but the response was slightly greater in nitrogen plots. So soil respiration was responsive to increases in soil temperature under nitrogen fertilization. Now, this would be great if this was the only story we had, but it continues, as you might imagine. So here's a nematophore. So those of you who don't know what a nematophore is, this is what they look like. They're these little tinsel roots that stick out of the soil, and they're responsible for gas exchange. There's, of course, debate about what type of gas is most prevalent. But one thing I want to point out to you is the y-axis. Nematophore, the rates of gas flux in nematophore, this is a 0.5, this is a 0.3, right? So the overall rate of gas flux through these nematophores, the capacity is fairly high. Okay, so what did you find here? Well, not the same task. Nematophore respiration was stimulated by nitrogen in the dry season and phosphorus during the wet season. Doesn't match what the soil is doing. So it's kind of interesting to think about the 2 different responses, how it's manifesting in the live roots or aerial roots versus in the soil itself when you have a large microbial community that's being affected. And here are the temperature curves for that too. So nothing related to nitrogen, but the control and the phosphorus, of course, have this response. It's interesting to think about this a little bit because all of our treatments are technically controlled. We're really comparing nitrogen to control and separately phosphorus to control. So it's hard to not think about nitrogen versus phosphorus, although it helps with interpretation, I think, a little bit. But if you look at this, there's no responsive nitrogen versus control. So just as a summary, limiting nutrient appears to shift between nitrogen and phosphorus and stimulating soil and live root here meaning nematophore, with nematophore respiration consistently higher than soil respiration. And respiration rates are higher than other mangrove forests. So this is what's interesting. If you look at all the data collected from mangrove forests, really throughout the world, these rates are much higher. So one question we had is, well, maybe we're not seeing phosphorus and nitrogen, clear, super responses yet. Maybe it's because there's this background loading that had this system responding. I mean, there's so much phosphorus in the soil now, maybe additional phosphorus doesn't matter that much. So that's just one thing that's in the back of our mind. So the second part of the ecosystem metabolism question has to do with the leaf scale. So this drawing here is, and this was done by Eric Ward, who's a scientist at the Wetland and Aquatic Research Center with me in Lafayette. And so just wanted to let you know, this red circle here just tells you this is what Natalie worked on the previous slide. He wanted to look at, okay, if we're going to fertilize these plots, then what's the very first thing that's going to respond to it on a tree? Well, it's going to be the new leaf. So he wanted to look at these photosynthetic characteristics of these leaves because photosynthesis is highly controlled by nitrogen and phosphorus. So this is kind of one way to look at whether or not these trees were taking it up, but another way just to see what the effect may have in the short term. So he looked at two variables. Both are a function of light saturated photosynthetic rate. So this is photosynthesis, carbon going into the leaf, versus the internal leaf CO2 concentration. And he measures this on this infrared gas analyzer in the field. And he established two rates, basically a rate of change between the two, which he terms VC max, and a rate of sort of an efficiency between these two factors, photosynthesis and concentration of CO2 in the leaf at light saturation. So, you all know that when photosynthesis, when it hits light saturation, it asymptotes. So these two variables are really responsive. And the literature tells us it's super responsive to nitrogen and phosphorus. So he wanted to know what was going on with these mangroves. And here's what he found. The same thing for both. So what you're seeing here is nitrogen fertilizer. It stimulates both of these leaf characteristics relative to phosphorus, but neither nitrogen nor phosphorus stimulates or reduces the response relative to the control treatment. So again, you have this situation where we can compare nitrogen with phosphorus and say, well, nitrogen is doing something to the leaf photosynthetic capacity, but we can't really say it relative to the control, which is how the experiment is set up. So this suggests that nitrogen and phosphorus additions have different effects on leaf photosynthetic capacity. And also remember that these small effects can change the structure of the forest or the tree and really have a manifest in different ways through the way that individual trees respond. But you might imagine you need time for this. 3 years might not be enough time. So the next step in the ecosystem metabolism is the tree scale. This is work done by Haley Miller and Jamie Duberstein at Clemson University. And this is always fun work to do. So they set up sapflow studies during two growing seasons, actually one winter period, and looked at sapflow of red mangroves and the black mangroves. These figures here are from the red mangrove. And I just wanted to show you a few things. You can't really differentiate any differences amongst treatments based upon these figures. But what I wanted to show you is that they installed sap probes at 5 millimeters into the tree, which is really, really shallow. It's the very outer sapwood. And they did this because they wanted the sapwood to represent new growth, especially in the second year. In other words, growth that actually occurred after the fertilization. But if you look at the solid red versus the open circles are sap flow at 15 millimeters into the tree. So it's still higher as you go into the tree, but there's some sap flow in this very outer band. Now they take these data and they combine it with sap flow sensors that have been inserted at even deeper depth. And they're able to determine individual tree water use, which is a lot easier to visualize in your head. And this is what they found. So this is individual tree water use in liters of water per day. And year 1 and year 2. Now, these are different trees in each year. We replicated in time, and we replicated in terms of the trees we're using. The only real trend that we find here is in nitrogen-treated Avicennia marina, black mangroves. You see that it sort of differentiates here only from the phosphorus, but in year 2 it differentiates from both the control and the phosphorus treatment. That's the only real thing. Lots of variability here. We don't know what it really means. It's not a super strong relationship yet. And mentioning here also that red mangrove had very little response. This looks kind of promising. The nitrogen and phosphorus may be suppressing it a bit, but then you look at year 2 and you just don't see that same effect. Also, I'll mention that all of these studies were conducted in the basin community. We just didn't have the same distribution in the fringe community to look at. I don't think we did in the fringe, but anyway. So nitrogen fertilized black mangroves tended to have slightly higher individual tree water use. Nitrogen fertilized trees of Avicennia had higher relative sapflow rates at a depth of 5 millimeters versus 15 millimeters. So this is important. We saw most of the response in the outer sapwood bands that are associated with newly produced xylem tissue since the fertilizations began. And this is a big thing, right? Because this is equivalent in a way to what Eric documented in the leaves, is that the new material seems to be responding to the fertilization. And so this is what I mean. So this is a cross section of a tree. This is not a mangrove, because I want to show you the growth bands here. You can't see growth bands very well in mangroves, but you see this part here. The green represents the 0 to 5 millimeter band, the new growth. And of course, water is used probably to about here or so in a mangrove forest. Okay. All right. So the final variable that we looked at was this response to soil surface elevation change. We use surface elevation tables. I think most of you are familiar with these, but what they are, it's a way to measure soil surface elevation change at repetitive locations over a period of time relative to a deep datum that was installed to refusal. So these are to survey. We install surface elevation tables to survey standards. So really, you have minimal amount of movement here. You can account for it based upon comparisons with tide gauge data. But we basically look at the soil surface movement over time. And we do lay an accretion plot as depicted here in the photo, but I'm not going to talk about that. You can really dive into what's happening to subsidence, zone expansion, all these different aspects of the surface elevation change. But the bottom line for us is surface elevation change relative to sea level. So with that, this is what we found. Y-axis shows you surface elevation change in millimeters per year, basin on the left, fringe on the right, control nitrogen and phosphorus. The one thing I want to point out though, the NS refers to not significant. What that tells you is that the repetitive measures over time in the control fringe, repetitive measurements over time in nitrogen fringe, do not differentiate from 0 statistically. So basically, these are 0. They certainly statistically are 0. And this is fairly high rates of surface elevation change for a mangrove forest. So we found that basin control treatments were higher than fringe control treatments. Basin nitrogen treatments were higher than fringe nitrogen treatments. So in other words, what I'm saying is basin control and basin nitrogen are both higher than 0. And that's the response that we found here. So nitrogen and phosphorus didn't have an effect. So we thought, well, wait a second. What about maybe we could test this idea that it's the nutrient loading, the background nutrient loading that's really causing these systems to differentiate. We thought, the Caloosahatchee River has been delivering nutrients for a while. So let's look at the long-term rates of accretion versus the current rates of accretion. And fortunately, we had the data from Judy. That she differentiated a 50-year trend versus a 100-year trend. And I'm superimposing that upon the controlled treatments for the basin because that's the appropriate comparison. And you notice I don't have it for control treatment and fringe because of the issues we have with dating. I'd really like to redo this because it may be that the fringe is actually the systems we should worry about. But you'll see that there's nothing really different between background rate of accretion over 50-year and 100-year periods versus the current rate. So there's nothing special there. Again, it's unknown for fringe at this point. And just to give you context, this 3 and this 3 are somewhat aligned. These are the data from the wider Caribbean, Florida Caribbean region. Median's about 3, mean's a little bit lower, and there's a lot of places that are actually losing elevation. So what we can conclude is that there's no obvious sign that surface elevation change has been compromised just yet in the basin. Again, fringe, I'd like to learn more about the fringe. I think we all would. Okay, so result summary. The mangroves at Ding Darling currently hold a lot of carbon. We want to hang on to that carbon, and you know, we have a small concern that the region is not recovering as we'd like it to from the 2004 Hurricane Charlie. And you can kind of see a lot of structural damage still after the storm, and now even after the storm. Experimental loading of nitrogen and phosphorus influenced rates of soil and root respiration, but in different ways and in somewhat inconsistent ways that are a little bit difficult to interpret. Small differences in leaf photosynthetic traits were detected. So we know that the trees we're pretty sure that the trees are picking up the nitrogen and phosphorus. So the question about was the fertilizer delivered properly, I think can sort of be answered a bit here. So there's some real-time adjustment to higher nutrient loads. Individual tree water use was influenced in one species, Abyssinia germinans, most of it in the outer sapwood band since fertilization began. Not much influence on the red mangrove. And I should mention too that we didn't have enough of the white mangroves, Laguncularia, in many of our plots to really focus on that. We do have growth data from white mangroves, but we don't have a lot of the physiology work on it because it just wasn't largely represented in the systems we're working on here. Surface elevation change was higher in basin mangroves relative to fringe mangroves, and the rate of surface elevation change in the basin mangroves really is indicative of a system that's expanding, that has a lot of root zone expansion. It's a high rate of surface elevation change that we need to sort of learn to interpret. But it wasn't distinctive. Rates of change weren't distinctive versus pass rates or by treatment, nitrogen and phosphorus. And so we can sort of conclude in a way that while there is some growth in metabolic shifts associated with experimental nitrogen and phosphorus loading, growth in surface elevation change may already be maximized because of the previous loading. We don't really see this. It's not, we don't see this strongly in the basin curve, but maybe in the fringe curve. And so we can kind of think about it like in terms of, initially we were thinking about looking at sea level rise modeling based upon a nitrogen fertilization treatment versus a phosphorus fertilization treatment and creating different vulnerability maps. But we're not finding the differences that really justify us doing that. So the next step that I'll show you now is that what does this mean for the future of mangroves at Ding Darling National Wildlife Refuge with continued sea level rise? So we developed, this was done by Karen Thorne and Kevin Buffington at the USGS Western Ecological Research Center. And they've been working with the warmer landscape feedback models for a while. And they've been recently applying this model to mangroves. And it's really, really cool. One of the first things that Kevin does is he'll come into a site and he will do LiDAR corrected digital elevation models. So he updates the digital elevation models for the region or the area that he wants to model. And then he calibrates the model for that particular site. And I'll show you, these are the input data sets. All of these data came from Ding Darling except for the root size characterization. We didn't have the data available yet. We had the data being collected, but these came from Everglades National Park, not that far away. And so everything is superimposed upon this elevation. Now, this is mangrove colonizable elevation. So you'll see in a second, the area is a little bit more than the 1,000 hectares that I showed you earlier, because there's areas it's got to account for accommodation in terms of movement upslope, as well as there's probably some unoccupied areas downslope that's included in this particular digital elevation model. Okay, and there's just a few curves. So again, red and black mangroves are the biggest story here. And there's just, there's these calibration curves that are inputted in the model. And then Kevin does a series of field calibration. So the different color lines here are the model outputs, and these dots are the data. And many of you know how modeling works. You try to tweak those to match each other over time because you want everything to be field verified. So he's done a lot of work to try to do this. But there's work to be done still before this gets published. But here's the preliminary result. So at a low sea level rise scenario, so this is 53 centimeters of sea level rise. I believe this is from the year 2000 to the year 2100. And so it's a low rate of sea level rise, lower relative to some of the other rates. And you'll see this is, of course, current distribution 2020, so you see 2040, 2060, 2080, and 2100, where you start seeing some of the interior mangroves starting to disappear by the year 2100. And what's interesting, well, I'll get to that in the next one. So I'm going to now show you the medium sea level rise. This is medium rate of sea level rise, but this is still a pretty high rate of sea level rise, so don't freak out. It's just that one of the reasons why you do this model is to kind of figure out where your threshold and tipping points are. And what Kevin determined is that the year 2050, slightly around that year, is when some of the tipping points in this mangrove forest may be reached, with or without the nitrogen and phosphorus, additional nitrogen and phosphorus loading. And actually, you see this in a lot of models throughout the world that somewhere around 2050, the capacity for vertical adjustment becomes somewhat compromised. Okay, so I'm not going to show you the high scenario because it's kind of scary. But here's the linear version of low, middle, and high scenarios. And you'll note here that this is 1,500 hectares, not 1,000 hectares. And this is because this is mangrove colonizable. Some of this area is not necessarily mangrove right now, but can be. And so there's a lot of calibration that he did, that Kevin did, and Karen did, and that there's more to be done. Probably a few more months left. And the result is that under low sea level rise rate, the model warmer projects minimal loss of mangroves. With mid to high sea level rise rate, it's substantial loss, with potentially rapid declines after 2050. So that's just sort of the first run through of all these. So the next step and the next tool that I had mentioned is this ecosystem services modeling. So I mentioned this because both of these are different types of models. One is to talk about vulnerability. The other one is to talk about value moving forward. So this was done by Emily Pendilli. She's based in our headquarters office. And she developed this really neat, this looks pretty basic, but there's a lot put into this these sort of connections here to try to figure out all the elements that you need to include into valuing mangrove wetlands. Most of her focus is on climate regulation via carbon sequestration, as well as recreational fishing and wildlife viewing. I already mentioned that wildlife viewing is huge for the refuge. And the values are pretty incredible, actually. So climate regulation via carbon sequestration, she gets a total economic value of CO2 stored by the refuge of about $55 million. Now remember that this is in comparison to all the values that carbon brings, not just the sequestration of carbon versus carbon markets. There's a way to buy and sell carbon right now. It has to do with climate regulation and all these other consequences of not having this, this CO2 imbibed in the soil or in the above ground biomass. So it seems like a lot, but it's real, and it matches a lot of what you see globally. If you look at wildlife viewing, fishing, economic value, this entire refuge is worth about $85 million, at least this economic value assessment based upon the ecosystem services analysis, about $85 million, which is pretty amazing. And it kind of makes sense too, because those of you who know Sanibel Island know that it's a super expensive place it's, I mean, just think about the value of land there. I mean, you know, it's through the roof. So it all kind of makes sense. So our next step, the only other thing I want to say, and I'm about to close this out, is we are in the process of, the background of this is we were going to set up an eco-variance tower initially, but then what happened was we had a sort of a scare in losing the money. So we decided to divert that money from Land Carbon to a different project and give them another tower or help them run their current tower. And because we thought we were going to lose the money. Well, we didn't lose the money. And then we set up all this work, and then we lost the money. So we didn't have an eco-variance tower set up. But we have this really unique modeling approach that Eric Ward, Jamie Duberstein, and I are looking at. So basically, if you look at the amount of water that a tree uses and a stand uses, if you can calculate the amount of CO2 those trees need to use that amount of water based upon the efficiency data that we've collected between canopy and water use. And this was successfully applied recently by colleagues of ours in Xiamen University in China. So we know we can do it, but we haven't done it yet. But it's a way to kind of get at eco-variance data without actually using eco-variance data. And the idea was to really split these carbon budgets up into nitrogen, phosphorus, and no future loading scenarios. We might still do that, but only if we get the right significance in our, you know, if we do decide that there are significant effects. Right now, they're hard to disentangle, some of them. And then finally, just want to thank everybody for being here. Thank the U.S. Fish and Wildlife Service for being an incredible partner, not only at Ding Darling, but also at other refuges that we work on in Southwest Florida and actually in the Southeastern United States. They're a phenomenal agency research partner for us. And if there's any questions, I'm happy to take them.
Ken, thank you so much for a fabulous seminar. Thank you so much. We have some time for questions, so feel free to ask a question. You can raise your hand through the participant box. You can also unmute yourself and ask your question or type it in the chat and we will share it for you. So thank you. Please type away. Okay, Catherine has a question. Wonderful.
Hi, Ken. I enjoyed that very much. Do you have any idea of how the inputs from the Caloosahatchee will change over time with climate change? So is there likely to be more or less rainfall that might affect this?
Yeah, no, I don't. And you know, that, and also just the efforts to restore some of the hydrology into that area, Southwest Florida in particular. I really don't. I did an analysis at one point for 10,000 Islands Refuge, previous antecedent rainfall versus current rainfall. And I want to say it decreased, but I forget. I didn't do that same analysis for this area though.
And can I tag on to that question? This is Carrie. So yeah. I was going to ask a similar question, but also, are there any regulations or ongoing BMPs or practices that might reduce nutrient loading in that watershed?
Yeah, I don't know. I know that the fact that it's getting serious headlines, and from my understanding is that it's one of there's 4 big rivers that come out of Lake Okeechobee, and it's sort of the one that's less regulated or unregulated, and of course it's causing a lot of the issues potentially. So I would hope that some measure of change is in the future. I don't know what drives that though, and what I mean, obviously these aren't the data sets that would inform that. It would be, like you said, the amount of water versus the amount of nutrients in the water. We were just really shocked at the amount of phosphorus in the soil. I've never seen levels like that before in my experience, and this includes places in China too, where you expect them to be high.
Ken, we have a question from Tom. It seems mangroves respond positively to nitrogen and phosphorus fertilization, but the eutrophication of the water in the estuary is harmful, is it not? And how can this discrepancy be explained to the casual observer?
Yeah, so I wouldn't necessarily say that nitrogen and phosphorus, that they respond positively to nitrogen and phosphorus. I think that I mean, not in this watershed. I think that there are definitely areas where nutrient limited mangroves occur, and when you fertilize them, they do better. But I don't think that necessarily, you know, if you put too much nitrogen or too much phosphorus in a mangrove, it could cause peak collapse. It could cause issues associated with you know, reducing the health of that system. And you know, in general, especially mangroves in Southwest Florida and in Florida in general, are somewhat adapted to low nutrient environments. So this is sort of a cause of concern for us because, in particular, that phosphorus level. So it's sort of an unknown what the long-term loading will have, unknown effect. But I wouldn't necessarily consider it a positive long-term response. You know, a lot of these experiments are 2 years in duration versus 20 years. So let's see, the water, and definitely the eutrophication of the water, as you're aware, is super harmful. So I think it could be a double harmful situation.
Any other questions?
Yeah, and I see that Paul Julian says there are indeed nutrient reduction targets and changes in water management is on the horizon. Thank you, Paul.
Yeah, thanks for that.
You're welcome. Yeah, there's definitely also nutrient or water quality specific target or projects being thought about to reduce watershed load to the estuary, which of course is going to affect, you know, the estuary further down in Ding Darling and all that kind of stuff too. And personally, I've been looking at watershed loading to the estuary for a little bit, looking at both the historic time series, but also in estuary conditions, and things are changing.
Yeah,
some good, some bad.
Yeah, thanks.
Yeah, that's great. I have another question if someone else, but I'll pause for a moment and see if there's anyone in the audience who has a question before I jump in. All right. So Ken, my question is, with regard to the modeling some of this mangrove extent going forward, and then, you know, and recognizing that included some colonizable land, are there any issues with land use change, especially kind of development pressures that might, you know, present obstacles to some of that migration or colonization? Have a sense of what the island, you know, land use is.
Yeah, well, fortunately, most of the area that's included in the modeling is protected as Department of Interior land. You know, definitely a shout out to the Refuge system, as well as the National Park system. You know, without that, my answer would be significantly different than it is right now. I do think, though, indirect effects of development need to be monitored. This is something we see all the time. I mentioned culverting, not putting enough culverts in, building roads, seawalls. You know, those can have effects on mangroves far down the beach or far down the estuarine fringe. And we're learning so that kind of stuff really needs to be looked at. So you could have development that affects the mangrove that isn't in the mangrove. But other than that, fortunately, a lot of this land is protected.
That's great. And I see that Jeremy Conrad has mentioned some additional data from the project that suggests that there's some reduced productivity in the fertilization plots compared to controls. So
yeah, and the photosynthesis data kind of alludes to the fact that greater phosphorus may be having an influence in real time. That, you know, again, it may need 10 years of fertilization to really see what we're trying to target here.
Right. Yeah, there's one more question in the chat. We are at the top of the hour, so I wanted to give folks recognize that folks have stayed with us this long. Let me just say real quickly, and then if people want to stay on, Ken, if you have another moment to address that question. Otherwise, we can follow up after the fact. But I guess I just wanted to wrap up to let you know that, well, first off, to say again, thank you very much, Ken, for that for this really great presentation. Huge amount of information here, and for some of the good discussion afterwards. And to let you know that we are recording this presentation, and we'll be posting it to our website, and I put in the link to where our science seminar page and where this will appear, and it will link out to our YouTube channel where we'll portal this as well. And by subscribing or by registering today, we'll subscribe you to our newsletter, which you are free to unsubscribe from, but hopefully you won't, where you can learn about upcoming seminars, etc. So let me say thanks everybody today. So whoever needs to jump off can do that. And again, Ken, thanks for this great presentation.
You're welcome.
And if you have another moment, you want to answer this last question.
Yeah.
Free.
Yes, thank you, Julio, for the question. Yeah, you're totally right. So we tried to standardize. I mean, we can't tell tree ages in mangrove systems, but we sort of tried to standardize the plots as best we could, visually by basal area distribution, et cetera. So there is. So if you have a pneumatophore necessarily that comes from a tree that's large, or even, yeah, I mean, there's a lot of characteristics of a tree that can drive that. And then there are certainly, not all pneumatophores are equal in being able to conduct gas or convey gas anyway. So we basically do the best we can to replicate the roots and set the plots up as uniformly as possible. And that's how we address that to the degree that we could.
Okay. And it looks like there's one last. I mean, the reason there was very little change in growth between controls and nutrient loaded plots is because of the already high levels of N and P in the soil. Did you compare the same growth measures at Ding with other mangroves and more oligotrophic areas?
Yeah, so that's part of what we're going to do for sure. In particular, sort of basal area, per unit basal area growth. And you're right, the phosphorus in the soil, it may be already responding to that. That's a hypothesis we have right now. I think that if you look at the surface elevation data, it isn't necessarily showing that just yet, but other metrics may, and definitely as a part of this study, you always compare what you find in one area with other places. I didn't include any of those data here. There's a wide range of productivity data. Some are lower than this, some are higher than this. From Southwest Florida.
Okay, great. Well, we'll call that a wrap then. And thanks everyone for participating, for joining this. And again, Ken, for a really nice explanation of some of this work. And
yeah. Okay.
Hope y'all have a good rest of your day.
Yeah, thank you.